All right, sorry for the slightly rocky road this morning. The moderator who was supposed to be in charge had to take off for a personal emergency, so I have stepped in. And I'd like to introduce our two distinguished panelists. First, Srikanth Ramaswamy, who is the group leader and senior scientist in the circuits section of the Simulation Neuroscience Division at the Blue Brain Project. So he works with Professor Dr. Henry Markram, Eliav Muller, Aydin Segev, and Javier de Felipe on understanding simulations of synapses. We also have joining us Arvind Kumar, who's a professor at the KTH Royal Institute of Technology in Stockholm, Sweden, and has done a lot of work in understanding how spiking activity propagates in neural networks. My name is Brad Wyble. I'm a professor at Penn State University and one of the organizers of the Neuromatch Academy. And with us also is Eric DeWitt, who is a postdoctoral researcher at the Champollion. Did I get that right, Eric?
I'm a research associate at the Champalimaud Research, which is the research arm of the Champalimaud Foundation. But the important thing to know is that we work in the building called the Center for the Unknown, and you can kind of dismiss everything else once you know that cool name. And glad to be joining you.
That's a great name. Okay, so we're in gallery view, excellent. So what we're gonna do is, first of all, go through some of the questions that folks are asking. And I would invite all of you to please feel free to talk amongst yourselves within the chat. We should have enabled chat with all the participants, is that right?
Yes, you just need to remember to select in your chat panel all panelists and attendees, otherwise it will only come to us and only we will be able to see your interesting thoughts. So share them with everybody in the community by selecting all panelists and attendees.
And Arvind, you were in one of these Q&As just a few hours ago, is that right?
Yeah, me and Eric, we both were there.
Right, okay, wonderful. It's early morning for me here in the East Coast of the US, so putting things together.
Mm-hmm.
I had the same experience this morning for hours, so I know what you feel like.
Okay.
So I want to emphasize also that the point of the Q&As is to get into the sort of deeper theoretical and philosophical issues related to the content, not as much the nitty-gritty of the specific tutorials. So we will try to answer those more fine-grained questions in text in the Q&A block. But the ones that we're going to talk about with the panelists are going to be the ones that we think of as sort of broader context.
Okay.
So,
I must mention that I wasn't part of the tutorial, so I'm not exactly sure what was discussed.
right.
I think to me, it's really more of a general overview. But,
Yes.
Yeah.
Yeah.
So it's touch and go. It's not really delving into the details.
This is, I think, totally expected, Srikanth, and you are here mainly because you have a very big expertise in detailed neurons at various levels. So that is something that I don't know if Eric has much expertise. I definitely don't know much about detailed models. So.
Yeah, I mean, I've been involved with teaching them, obviously, in a number of computational neuroscience summer schools, and I've worked with them a little bit, but my own area is reinforcement learning, where I get to abstract over all of that, which I enjoy. But I actually also really enjoy thinking about how we can go from these detailed biophysical models to reduced models. So one of the things I find interesting about these conversations is, in the same way we talk about what, how, and why questions, and we talk about Mars levels of abstraction, there's also sort of a, how do we go between, say, a biophysically detailed model with active ion channels and all of the complexities that you can imbue into a single, say, pyramidal cell model, and reduce that to something like a Fitsugo-Numo model or one of these simpler, more abstract models where we make some assumptions, but it gives us the power then to look at larger sets of networks and so on until we get to what we think of as simple rate models or the kind of nodes that are used in deep neural networks. So I I guess the reason why I like to go back and forth, part of it is I'm also an experimentalist. So when I stick an electrode into an animal to record from the neurons, I have to think about these things a bit. But also because I think trying to think about how we move back and forth between these scales is just it's a really important thing in computational neuroscience. And this is one of the areas where we can think about it very clearly.
I think that's one of the biggest debates in all of modeling, is at what scale do you implement these simulations? And there are some people who have extreme opinions. You know, some people think that you should do everything at the compartmental level. But, you know, it's not necessarily clear. I think that that's always the right idea for every model, because as you say, Eric, sometimes you want to sort of zoom out to the big picture.
Right. No, indeed, there is no one size fits all here. And I would like to point out a reasonably recent review from Chris Elliott-Smith. I think it came out in Current Opinion in Neurobiology some years ago. It's called The Use and Abuse of Neuroscience Models, or something like that. I think the moral of that story really is that there's no one size fits all. And the extreme levels of detail on one end of the spectrum and complete abstraction on the other really depend on the kind of questions you want to answer. If on the one hand, you want to understand why the third order dendritic segment of a layer 5 pyramidal cell is involved in modulating gamma oscillations, then you better model that, or you won't be able to answer that question. If, on the other hand, you have questions that are purely related to the network level, then possibly you could try and abstract some of these details. But one thing's clear in that it's becoming more and more important to try and come up with some kind of a reference with sufficient amount of detail that you can begin with, and then try and shave off the detail until you arrive at a level of abstraction that suits you, because then you have a reference. And this, I think, is gaining more and more traction in the field from where we were, like, I don't know, a decade ago, where detailed modeling and simplified modeling were totally tangential. But now I think there is more and more of a convergence, which, again, in my opinion, is what the field really requires.
Yeah, I think it would be a nice addition to the way that we write modeling papers, is to justify your particular level of scale. I mean, I have personally seen papers where there's an incredibly detailed compartmental model with all kinds of oscillations and things, and, you know, ion channels for a specific neural circuit that absolutely did not require them. You know, and I think sometimes modelers include extra levels of detail because it makes it easier to, you know, to talk about the paper in this deeply detailed neuroscience sense, but it really is a distraction in some cases from the specific core mechanism that they're really looking at.
Right, yeah.
I think one of the worries, if you go back and forth between actually conducting your own experiments and doing modeling, you quickly realize how few constraints we've actually firmed up in the experimental side, right? If we take a look at a complex model, normally in your experimental work, you can manipulate a few things. Maybe you clamp the neurons and you perturb it in a few ways, but there are so many aspects that we simply don't have the time experimentally to manipulate. And then if you use a very complicated, detailed model without good, you know, so strong priors from, say, background experiments, you just know that many of those assumptions will be unconstrained. And so then you have to ask yourself, was it necessary to put them in to explain the phenomena that you were, you know, looking at? But that's always a scientific question, I think. You know, it's not a right answer or wrong answer question. It's having to think back and forth.
Mm-hmm.
Well, we've got some great questions coming in. Let's start off with one here.
Right, sure.
So,
can I say something before you start?
Yeah.
There are very nice questions in these. I just went through them. And I don't know if we will be able to take up all of them, because some will fall under the category that they are too technical, specific to the tutorial. So I would suggest that those questions that are not taken up, could you please take them at Neurostars, and we will do our best to reply them there. I think it would be a pity to lose these questions. They're very nice ones.
No, absolutely.
it's a really fair, Arvind.
Right. So, I mean, there's one question that clearly gained my attention.
Go ahead.
So should I point you to the question so you could,
Sure,
you
yeah,
could initiate the discussion? So there's a question from Julia Denny on how does the physiology or models of neurons vary between species?
Yeah, so what's your
Right. So the short answer to this question is there are subtle variations, but by and large, the phenomenological features seem to be preserved across species. However, these also are dependent at a specific developmental timescale. So, for example, if we talk about synaptic connections between layer 5 pyramidal cells in rat or mice or young humans, young adult humans, where people with chronic epilepsy usually have to undergo a so-called lobotomy procedure to reach the nucleus of the epicenter of temporal lobe epilepsy to come up with some deep brain stimulation protocols. And as part of this procedure, a lot of the healthy tissue of the cortex is taken out. And some of this tissue can be resurrected to do the same kinds of experiments that we do in vitro in rodents. For example, making about 300 micron thick slices of mice or rat or even human now cortical tissue. And it appears that, for example, the fundamental features of synaptic transmission, like between layer 5 pyramidal cells, are strongly depressing in young adults, be it young adult mice or rat or humans. So it seems there is some kind of preservation of synaptic or even cellular properties across species, but this is really limited to one snapshot in development. And as animals, including human beings, develop, it seems, it could very likely seem that the balance of synaptic transmission, which is mostly in favor of depression in the younger animals, tilts more towards facilitation in older animals as animals mature. So long story short, yes, some of these properties seem to be preserved, but they're very age dependent.
Are you aware of any specific mechanisms that have disappeared in the evolutionary tree between species?
Yes, there are some specific examples, of course. Like for example, it seems that the dendritic arbor of a layer 5 pyramidal cell in the human cortex is obviously much longer than its counterpart in the mouse or the rat cortex. And apparently, what evolution has succeeded in doing is it seemed to have endowed like specific properties to subcompartments within this elongated dendritic tree of human pyramidal cells. So it seems that each of these subcompartments are self-sufficient to carry out their own computational role, as against the more compact dendrites of layer 5 pyramidal cells in mice or rat, which seems to suggest that these subcompartments, given that they could be more self-sufficient, could also have their own different forms of excitability because they have different expressions of ion channels, so on and so forth. So they could really function as distinct computational units as you move from mouse to man, probably.
Anything to add, Arvind?
No, I think this is something I didn't know actually.
Yeah.
I assume that these are all preserved across species and that's it.
Well, I mean, I think what we know is that, even though there's homologous structures and homologous, gene expression patterns, often the details are different, even in the mammalian species as you get far enough away. And of course, we have more diverse subtypes in some species than others. So even when you might have one common neuron that has, common genetic underpinnings and expressions, you may have a variation or variant that's in one species that's not another. I think to me, what's interesting is you can go as far away as, insects and mammals and still look at a lot of the fundamental properties of these single neurons and find that they're conserved, right? So to me, what's interesting is trading off between the balance of looking at what's been well conserved versus where there are changes and trying to ask why. Are those just sort of, non-selected adaptation that comes with being on that phylogenetic branch, or are they things that were really serving a particular purpose in the cognitive domain of that species, or the computational domain of that species?
Yeah, and whatever data that exists on human neurons, for instance, there what you see is more a quantitative difference, if at all, like the dendritic arbor is bigger, or the synaptic transmission, dendritic transmission speed is faster. It's more quantitative, not qualitative.
Yeah, I mean, to sort of extend the analogy to stuff we'll talk a lot about deep learning at the end of this week. You might think of that evolution does more work changing the hyperparameters than the specific subparameters as it tweaks organisms.
Mm.
All right, so here's a good question that I don't know the answer to. It's by Betsy. It's at the top of the pile here. Can we think of any examples where very detailed biophysical models have been necessary? I'll add the word necessary for modeling higher level cognitive function. Like a case where some like a specific compartmental modeling approach has played a key role in simulating some higher level cognitive aspect.
So what's a higher level cognitive aspect here? Gamma oscillations are a higher level cognitive aspect, okay, because they seem to be a direct readout of attention or concentration in animals, for example.
Yeah, that's a great example. Yeah, oscillation.
Or working memory.
Really, that arrives from that.
Yeah, I'm not aware of working memory, but yes, I do know that there are some examples of detailed biophysical models that have been used, not necessarily to specifically model gamma oscillations, but that have resulted in gamma oscillations as an emergent property while trying to understand, or while trying to replicate certain experimental studies in a network that was used, or rather in a network that was constructed with these detailed biophysical models. So yeah, I mean, the answer is yes, but I'm not sure that's really the expected answer to this question. Perhaps there's some more detail that's expected. I mean, I don't know.
Another example that comes to mind is NMDA channels. If we think that, so the specific property of NMDA channels is that they require both pre and post-synaptic depolarization in order to potentiate, or sorry, in order to open, and then that leads to potentiation. And so these NMDA channels have been thought to play a key role in the formation of stable long-term memories.
Another place where biophysical properties have been super important is epilepsy modeling, where people have been focusing on specific ion channels. Like, I don't remember all the details now, but I know that in that field, there are very detailed models as well, all the way like from Roger Traub in late 90s, I think. So yeah, I mean, you have to define like what higher order, higher level cognitive function you mean, whether it is working memory, gamma oscillation, or something as big as epilepsy.
It's funny
or
because
learning and memory, like what?
Yeah, I mean, I think all of these are great examples, but in some ways I would push back and say none of them seem to be what maybe is being implied by a higher level cognitive function.
There's a very interesting suggestion by Jose Guzman, who says that we could think of pattern separation or pattern completion as a higher level cognitive function.
Well, sure. Do insects do that? For example, in their olfactory system?
Yeah.
I think I would invert the question and ask, if you look at the human brain, or even, I don't know, I have a pair of cats, they do incredibly complex behaviors. And I bet you that at the end of the day, a lot of the biophysical details of how various nuclei are wired up are going to be critical to understanding how they do those. The fact that we don't understand it yet doesn't mean that it's going to be unimportant. And maybe it'll be important for the efficiency of the way the computation is implemented. How do you get that computation into a skull and have it operate on a few bananas, as happens with, if you're working with monkeys? It's not clear though that, it's not clear to me that we have higher level cognitive functions modeled by abstract neural networks either, right? Just because a deep network can identify a cat from a dog, is that a higher level cognitive function? So I think part of the problem here is we think we have an intuitive idea of what a higher level cognitive function is. I would agree with the NMDA example. I would agree with all of these examples in some sense, but at the same time, I think most people want it to be, how do we produce language or something like that? And the answer is we simply have no idea how the brain does that.
Right. Yeah, and I think that, the toolbox that we have is enormously flexible, right? And so, as Eric says, I agree completely that, you could potentially implement something in a zillion different ways using relatively abstract neural principles. So it's not necessarily the case that you would need this particular mechanism in order to do that particular function. It's just that this is the particular way that the evolution has settled on as a solution.
I'll give an example from my domain, which is we think that gap junctions between dopamine neurons help them synchronize, that there's something about that. And in fact, what reinforcement learning theory says is you want a relatively common, uniform, scalar signal that's going to be used to inform a bunch of changes in the rest of the brain. Now, is that necessary? Is that the only way to do it? In fact, we see that there's variation across those neurons when we look in more detail. So I think the answer can be this is informative and helps us to think about it, and maybe actually going in and looking at some of these details when the larger space of that more abstract framework is worked out will become quite important. But we may not just be at the point where we have precise questions that allow us to know, which particular details are important for which particular aspect of a higher cognitive process.
Right, so we've got a few questions here now on the EI balance, right? The balance between excitation and inhibition. So David Wolf, for example, is asking a question about the EI balance. How does the brain implement that balance biologically? And why is it, for example, that inhibitory interneurons are often very high frequency? So why specifically would we expect those neurons to have, 200 spikes per second? Any thoughts?
Well, it's not that inhibitory neurons. Well, okay. So roughly about 30–40% of inhibitory neurons are the so-called basket cells in the cortex. And of these, it's not that all of them are fast spiking. Yeah, some of them are, but some of them aren't. And then there are also the vast majority of other non-accommodating interneurons, which are definitely not the high frequency or fast spiking interneurons. So I mean, fundamentally to me, this is really a seesaw game or a way to balance the yin and the yang. But I mean, again, it's not that, as I said, I don't think interneurons are often very high frequency. I mean, there's a proportion. Some of them are fast spiking, some of them are stuttering, some of them are regular spiking. But yeah, I mean, I think the more interesting question is, so how's there a balance between E and I, despite this huge diversity in the firing types of interneurons, as against the monotonic accommodating firing pattern that's pretty much extended to all pyramidal cells, be it in the cortex or the hippocampus. So I think the fundamental question is how is there this a balance between yin and yang, given the fact that there's an overwhelming majority of excitation or this monotonic accommodating firing pattern of excitatory neurons in the brain. So to me, I mean, that's the fundamental point. And one way that the brain probably does this, that goes awry in disease, is the function of neuromodulators that Eric has already mentioned. He alluded to dopamine, but there are the big 5 neuromodulators. So there are another 4 in addition to dopamine, which are probably involved in a potpourri of cognitive functions, right? So there's histamine, there's acetylcholine, there's noradrenaline, there's dopamine, and there's serotonin. And these neuromodulators are working in tandem as we're doing all kinds of cognitive tasks. So one neuromodulator is setting the stage for the network to set its balance either in favor of E or I or whatever, and then another neuromodulator takes over. So there's this dynamic reconfiguration, there's this dynamic sculpting of the network that's caused by these neuromodulators, which are bringing about these changes in excitation and inhibition, simply because these neuromodulators have a differential effect on populations of neurons. While the same neuromodulator can enhance excitation in some neurons and their synaptic connections, the very same neuromodulator can cause different types of neurons to have more inhibition. So to me, neuromodulation is really a key aspect in controlling EI balance in neural computation. I mean, how these go awry, for example, when neuromodulatory signaling is disrupted, is directly translated into an imbalance in excitation and inhibition that's very prevalent in all kinds of disorders.
Yeah, and there are also sort of microscale anatomical solutions to this problem too. For example, you can have shunting inhibition delivered onto dendrites, right? And that shunting inhibition will have an enormously strong impact on basically amplifying the effect of inhibition across an entire dendritic arbor. So even you know, if you sort of like, it's not as simple as just counting up the number of excitatory and inhibitory synapses, right? You have to think about exactly where do those synapses land on the dendritic tree.
Sure, yeah. I mean, and to add to that, well, sure, when you have synapses from a specific axon forming on the dendrites, then there's some method to madness. But neuromodulators make this more complicated because it's believed that by and large, neuromodulatory signaling is non-synaptic or through volume transmission, where there's an axon that ends up in some part of the brain and just sprays neuromodulator, a specific neuromodulator that targets all neuronal components in its vicinity. So in addition to what Brad just said about shunting inhibition and how specific this is, depending on where a synapse sits on the dendritic tree, its location, this is also continuously being enhanced by neuromodulators that are released by volume transmission. So it's really a zoo out there.
Yeah, just a couple of more things to add to this. So what Srikanth talks about is somehow a modulation of the EI balance, but already at the hardware level, it seems that there's a lot of evidence that total amount of excitatory drive is balanced with the inhibition. I mean, clearly, a brain cannot work if it has excess excitation. It will be unstable and it will die out. There are various ways. So firing rate of interneurons is definitely one player in that. Their location on the close to soma is another player in that, which helps balancing. Remember, the question arises because at least in neocortex, we have 20% inhibitory neurons and 80% excitatory neurons. Right? Already at this level, we have a huge imbalance, and that somehow has to be balanced out. In theory papers, we often say that it gets balanced by scaling the synaptic strengths, but that simply means that you assume that inhibitory synapses are perhaps closer to the soma, or that neurons spike at a higher frequency. There are also papers which show that somehow if there is an excitatory synapse on a dendritic arbor, then it somehow increases the probability of formation of an inhibitory one instead of excitatory one. And very recently, I think a couple of months ago, there was a paper from Rob Fromke's lab where they showed that there is some sort of plasticity, which sets the EI balance as a target value. So it seems that every neuron has some intrinsic EI target ratio. We don't know why it should be that, but looks like plasticity rule works towards that value. So there are many ways how you can achieve this. And if I may continue, I think another question by David as well is about brain diseases and why it is such an important player in this game. So stability is definitely one issue. That if you don't balance E and I, then your networks are unstable, and before you get these other diseases, you will end up with epilepsy. But let's say that you are not so unstable. Then what might happen? So EI balance serves a lot of functions. Tomorrow in the lectures and also in outro by Ken Harris, which Ken is that?
Harris, I think.
Miller.
No,
Ken
Miller.
Miller.
No, sorry. Yes.
Sorry, there are too many Kens. So Ken Miller will talk about different aspects of excitation inhibition balance, how they help you compute something. So by varying EI balance, you change your system, let's say from what he will describe as inhibition stabilized dynamics to some other form of like, what is it called? SSN. So by changing that, your network changes its behavior, its gain function changes, the size of fluctuation in the network activity changes, it becomes more or less correlated. And I think all those phenomena, that translates to, let's say, schizophrenia or PTSD or autism and all these things. So one very easy way to relate EI balance to diseases is that one, it will change the gain of your network. Second, it will change the excitability. It might also influence, let's say, duration of working memory. It will change the frequency of gamma oscillations. It might create spurious patterns, and so your pattern separation or pattern completion ability is impaired. And this is how this very core property will have like effects all the way up to behavior.
But touching back on the point about why would this general principle give rise to such specific diseases? It's a really good question. And I think that the first question to think about with respect to that is, is it really that specific a disease? So like schizophrenia, for example, like in many cases, what we think of as a specific neurological condition is really an umbrella term that encompasses a wide range of different specific manifestations. So to some degree, we end up being guilty of sort of like inappropriately combining together a lot of different things under one label. So that can make it a little bit difficult to answer that question right out of the gate. And I think that, whether or not something is, sort of fundamental as EI balance would give rise to this wide range of different specific diseases, right? Like why might EI balance play a role in schizophrenia, or why might it play a role in autism and epilepsy? Like those are, sort of distinct kinds of manifestations. And why would you have this one principle underlying mechanism being the causative factor in all of those different things? And it obviously we don't exactly know the answer. These are speculations that people think about, but it's perhaps one way to think about it is that if EI balance were not optimal in a specific individual, it makes the system unstable in such a way that it might exhibit a wider range of different behaviors, right? And some of those might manifest as schizophrenia, some of those might manifest as autism. So it's just something to think about as being sort of away from the normal in this fundamental respect might give rise to a wide range of different outcomes.
If I may bring Mar here very briefly, Eric, just 1 minute, sorry. So Mar talks about these three levels, right? The algorithm, the problem level, then the algorithmic level, and then the implementation level. So if you think about EI balance, that's a problem at the implementation level. When we think of these diseases, those are more like at the problem level or at the algorithmic level. So what we need is this mapping between the implementation level phenomena that arises because of EI balance what I was referring to as, let's say, gain function changing or synchrony changing in the network. What it does at the algorithmic level is that, for instance, if you have high synchrony, then your capacity to carry information is decreased. Now, what kind of problems it will lead to next level, right? Those will be something that we will call as diseases. If it increases or decreases my working memory capacity, if it changes my way to balance the internal predictions of the brain and the sensory evidence. So my Bayesian calculations are messed up. So I may end up with either schizophrenia or ADHD. So this is how I think we don't really have a good answer, but at least this Mar way of thinking tells you how to translate these things.
Yeah, I mean, that bridges with what I wanted to say. So I just pulling on those 2 points. We have to think about the fact that a fundamental mechanism like this, which was sort of identified as being critical to stability in the system overall, so we know it's important, it could be disrupted in different locations within the brain differently across individuals. So a single kind of disruption, unless it was exactly the same uniformly, could lead to different outcomes. But moreover, those disruptions, if they happen at different points in the developmental trajectory of an organism, the organism adapts, and sometimes this can lead to a feedback level, not at the level of within the cortex, but as a feedback level in terms of the way it builds up its representations of the world and its responses. And so for humans, that means that perhaps some of these things, if they happen early in our development, end up creating one trajectory towards something we will call one psychiatric disorder. And if they occur a little later in development, you know, when you've already established some of the fundamental representations, maybe it'll end up, you know, going into a different psychiatric disorder. And the other thing that was related to what Srikanth was saying earlier, that there's neuromodulators on top of all of this, right? And so maybe you have EI balance that's okay, say, in terms of the sort of overall distribution of synaptic plasticity and their rules. But if your neuromodulatory tone is off, then at times you may have EI balance that's damaged by, you know, low tone of some set of neuromodulators that can be brought back with pharmacology, for example. But I mean, at this point, I think most of these things are speculation. It's just that the complexity of this relationship is not just because it's a single phenomena that we should have a single outcome explanation, but it's a single phenomena that then has to be intersected with these other kinds of factors.
I want to just close out this discussion with a specific point to remind us all that we're not clinical neuropsychologists, and you know, when talking about diseases of the brain, so-called diseases, you know, it's important to recognize that there's a wide range of different sort of possible instantiations of neural function, right? And so we sort of draw arbitrary lines and say these are disorders and these are not. And we need to be extremely careful that we're not inappropriately labeling things as diseases or disorders when actually they're just different aspects of the way that people can behave. And, you know, it would be great to dig into this discussion more deeply. I think we would really want to have a clinical neuropsychologist on the panel so that they could correct, you know, and help us to understand more about the specific behavioral aspects of some of these disorders.
Sure.
So I'm going to continue to scroll through the excellent questions here. If any of you folks on the panel if something catches your eye, please feel free to chime in. Had one here that I was going to bring up next. Let me see if I can remember which one it was. Okay, so Rawan Al-Subay brings up, and I'm sorry for mispronouncing that name, asks a question: if we can elaborate more on the implications and difference between nonlinear and linear integration in terms of increased storage capacity. So for example, when we talk about increased storage capacity, what's being increased biologically and how? Is this a question that resonates with anyone? And the answer may be no, and that's fine.
Well, I guess I'm still trying to see if I understand exactly what they're going for, because here it's a question of, well, I'll give you one example that's not quite to the point of this question, but maybe it will speak to it in some way. So there's been a lot of work in the last 5 to 10 years on nonlinearities within the dendritic arbor. I brought this up this morning in response to a sort of similar question. And those nonlinearities allow for very complex computations, up to the point where people are actually making the claim that you could think about the dendritic arbor as something like a small deep neural network. Because it has these active components, active channels, those nonlinearities within the dendritic arbor itself can perform computations, and we know that we can insert and remove ion channels within the dendrites based on activity, and so you could sculpt those computations. And now I've had a discussion with some of the researchers in this field where I think to myself, this is either an incredibly beautiful way to expand the computational capacity of the brain, where now we not only have to think about these kinds of linear, nonlinear cascades at the level of neural networks, but we also have to do it within individual pyramidal neurons. But there's an alternative approach to thinking about the complexities of the dendritic arbor, which might use exactly the same machinery, which is all of those wonderfully complex nonlinearities are actually there to make the processing that's done at the soma appear to be a linear summation with a single point nonlinearity, and a point nonlinearity that might be very stereotyped in some form. And why might we want that if we were evolution? Well, the neurons downstream that are trying to understand the information that's passing through this neuron, if they want to try to invert this in some way and actually make inferences about the computations that are going on, it becomes a lot more complicated the more nonlinear the system is and the more nonlinear the properties of every individual input are. And so as a speculation, I think one of the questions we have to always ask ourselves, are these things that we're seeing there for the purpose of expanding the computational power? Or as for example, we were just talking about an EI balance, there's a whole set of neurons and a whole set of processes that are probably about stabilizing the system, creating homeostasis, allowing it to do the complex computation as it does in this very complicated nonlinear system, but actually by imposing stability. So, I mean, I don't know, I think it's a completely open question, and it's kind of interesting whichever side it falls out on. But I think in terms of increasing capacity, there's a lot of advantages you could get by adding more and more complex computational properties, say, to an individual neuron. But you also probably lose something, because that means all of the systems that are trying to use that information downstream have a harder job of trying to do the fundamental thing they have to do, which is sort of learn what the inputs are and do something useful with it. Because at the end of the day, we actually have to wire up this whole brain and make it function as a single unit. And the more unstable it is, the harder that's going to be in some sense. So that's just sort of one partial answer to that question.
Yeah, I think I would just quickly like to add to what Eric said right now, and to also tie together some of the terms that have been used quite a bit in this discussion. For example, NMDA that was mentioned before. I think Arvind mentioned about excitability and gain control, and then this question on nonlinearities. Again, I'll also bring back what I mentioned, the layer 5 pyramidal cell, because the dendritic arbor of a layer 5 pyramidal cell, at least in rodents, for example, is quite extensively characterized. It's very exquisitely arbored in terms of its detail, and also supports a lot of ionic mechanisms that are distributed non-linearly across the somatodendritic gradient. Like, for example, the famous IH current, or the hyperpolarization activated cation current, that's permeable to sodium ions, for example, that increase dendritic excitability, that make them more excitable. So what some of these nonlinearities could actually do is by having a certain pattern of distribution, they could actually lead to an increased synaptic drive that, as Eric said, could lead to a lot more different paradigms of computation. And I briefly mentioned about the existence of these subcompartments in the elongated dendritic arbor of a layer 5 pyramidal cell in the human cortex. what these nonlinearities could be doing in these elongated dendritic arbors of human pyramidal cells is to really endow these subcompartments with very specific computational abilities, simply because the distribution of some of these channels is nonlinear. So if you take some compartment that's probably somewhere on the distal apical dendrite against one compartment that's somewhere on the proximal apical trunk, then by virtue of the fact that this one ion channel mechanism, I mean, there could be many more, but just to keep this argument simple and tractable, the distribution of this one ion channel mechanism in the proximal part of the dendrite compared to the distal part of the dendrite, given that it's more shallow in the proximal part and more steeper in the distal part, could make a whole world of a difference, because it's simply increasing the synaptic drive at your more distal part compared to the more proximal part, which could open up a whole Pandora's box for computation, which we have no idea about. I mean, I think we're beginning to appreciate substrates of why some of these ion channels are distributed the way they are, why there are nonlinearities, and why there are certain forms of distribution. Simply put, I think there are broadly three forms of distribution of these mechanisms along dendrites. So either they can all increase in some nonlinear way along the dendrites, they could decrease in some nonlinear way, or they just stay constant along the dendrites. So each of these distribution patterns really has many implications to bring about different paradigms of computation. And we really at I think the tip of the iceberg now. We are slowly beginning to appreciate the computational implications of these diverse distribution patterns.
Yeah, this sort of leads into, I think, another question. Number 2 at the top of the list here, Amireza Farbakash. Again, I'm so sorry for not pronouncing these names correctly. Given the immense complexity of neurons, is there any way to model or capture these complexities, right? Can we even hope to do so? And if yes, what is the way? And if no, what are we hoping to achieve in computational neuroscience? So I guess another question is, are these computational details important, and what makes the brain so special? And I guess my sort of off the cuff answer would be, yes, in theory we can simulate any of these complexities if we know about them. There's sort of no limit to the depths of things that we can simulate. The question is really, what things do we know about, and in what cases do we need to dwell on those particular details? And again, this also relates to sort of what question are you asking, right? Are you asking a question about how specifically does the brain solve this problem, or how can we build agents that can solve these problems more generally? All right, what do you folks think?
Yeah, so basically continuing where you leave us, that I think we should understand that modeling has a scope. You model to answer a question. And if there is a bottom-up reason for a particular biological detail to be there, you should include it. Alternatively, if there is experimental evidence that this particular component may have a function, you have to include it. So, either there has to be a mathematical reason to include something in your model, or there should be an experimental reason for you to include this. Just because it is there is not the reason enough to include it in the model. At least that's how I would like to think. In that, I take a lot of inspiration from Freeman Dyson, who received this advice from Pauli, who said that with 6 parameters, I can fit an elephant and make it wiggle its trunk, right? This is the famous story. So more parameters you have, more freedom you have, and you can do whatever you like. But whether you understand it, why it is happening, no. And the purpose of modeling is to understand some phenomena. And therefore, the only reason to include more detail in your model is either mathematical or some experimental evidence. Experimental evidence, not that it is there. Experimental evidence that it may make sense to have it.
Yeah, for example, like we don't think that we need to simulate accurately the behavior of mitochondria, right? Like there's good reason to think that that is not necessarily something that plays a key role in the neural dynamics that we in particular are interested in. But if you were interested in, the specific outcomes of a mitochondrial disorder, then obviously that's something that you would need to include. The mitochondria are the sort of energy units that basically create ATP, which powers neurons, if that's a term that you hadn't run into before.
I think Srikanth is involved in one such project where they are really going down to the energetics in neurons, in dopaminergic neurons, where depending on how the excitability increases and the neuron doesn't have enough energy and therefore it dies.
Right. Yeah, it's a hypothesis. I mean, I didn't want to go down that route, but yeah. Again, it's quite an open-ended question. So it's just a hypothesis for now, and we have no experimental evidence. So
I want to bring up a simple example that I know Arvind knows. So Arvind Kumar done a lot of work on the computations you can do within a single synaptic compartment using molecular models effectively. And it's incredibly computationally powerful. So everything we just said about the complexity of active dendrites can be lowered to another level. And of course, there's another incredibly powerful computational machine in every neuron, which is the genetic regulatory network, which is itself incredibly powerful. And I think one thing to keep in mind is that all of these are going to be very, very important for us to, in some ways, come to an understanding of various aspects of the brain and various aspects of the function of the brain. But at least for me, when I want to think about what models provide us, their goal in some sense is to reduce complexity, to give us a heuristic for how to think about the fundamental properties that are important for a particular set of questions. So, when I want to think about, my answer about the dendritic arbors may have given my bias away. When I want to think about how you might implement something like, Bayesian marginalization in neurons, you want those neurons to have the kinds of properties that will allow somebody to do inference using a set of these neurons, and they better have some kinds of stability that will allow you to do that. And so perhaps then we want to assume that these active properties may be simplifying things rather than making them more complex. That's a goal, a sort of dream as a scientist. Or in another example, when I think about how the dopamine neurons work in reinforcement learning, I want to, until I can no longer do so, treat them as one simple scalar signal, which is a vast oversimplification. And yet it's incredibly powerful as a starting point. And so, I think one of the things that we always need to do is choose these abstractions in a way that makes it easier to communicate about what we think the function of some particular part of the brain is. The hypercolumn model is an example of trying to think about a more compact description of a chunk of visual cortex, or maybe all of cortex, or something like the basal ganglia, taking inputs from cortex. I know Arvind has worked on this, which we think of as having some computational functions that we might be able to relate to even more abstract representations like reinforcement learning. But some of those details may inform us when we run into a problem, when our higher, more coarse models break down. So this dialogue back and forth is because models are an expressive language that's hopefully simpler than simply redescribing the state of the brain, the state of every gene, the state of every atom and molecule, which is, that's a language of describing the brain that's not particularly useful. If you just sort of were able to list that whole catalog, I think this came up this morning, with the connectome of C. elegans, just having something that's at a level of description that's complete doesn't necessarily solve the particular question you're interested in asking. So rather than worrying about whether we can or can't model all of these details, I think we of course could in principle, but maybe that's not the point of the modeling exercise, at least not when we're trying to model some of the things we're interested in.
Yeah, to add to Eric's point, so I think there is somebody called Anders Sandberg who tries to estimate how much memory it would take to store all the necessary detail of the brain. And it turns out that it would be cheaper to just preserve that brain. So a good model of a brain is the brain, preferably the same brain. So that doesn't really solve the problem, however.
Right. likewise, every child is a model of the brain, right? And so it is 10 o'clock. We started a few minutes late, so let's just go out with one last statement on, I think, a very nice high-level question. And the answer to this might be it's not quite the right question, but let's see what we think. Antonio Felasconi asks, at what point is the comp neural field in defining what is the functional unit of the brain? Is there such a thing? Does that question even make sense? All right, does someone want to go first? So candidate answers, for example, could be the neuron, the synapse, the column, the hypercolumn, the cortical area, etc.
Again, I think the term the functional unit is probably more dynamic than static, and I think as you try to put together a bigger and bigger spatial scale of the brain, then this functional unit representation also blows up. Like I don't know. I mean, if you're talking of a cortical microcircuit, then yes, it's the microcircuit that's probably thought about as the smallest functional unit, which could be defined as an ecosystem, a collective ecosystem where different populations of neurons all reside together and interact with one another to give rise to some kind of computation. But as you're moving beyond this spatial scale, when you're talking of one brain region, then I think the whole notion of this functional unit becomes blurry and blurry. I think to some extent it ties down to the spatial scale in reference. Again, as I said, to me, the functional unit is a very dynamic term. So it evolves as the spatial scale also evolves. Again, just my 2 cents worth.
I'll go ahead and say and go next so that I'm not the last one speaking. I want to give one of you folks the opportunity. I think for me, we often think of the neuron, I think, is the sort of important thing in the brain. Like, how big is the brain? Well, how many neurons does it have? But really, if you think about it, you could make an argument that the number of synapses is the more important variable there, right? Like in terms of the complexity, because you could have, if each neuron had 10 synapses versus 10,000 synapses, the sort of computational scope of what that brain could accomplish, changes by an order of 100. So I think, or 1,000. So, I agree that the answer to this question of what is a functional unit really does depend on the scope at which you're analyzing the problem. And we have a tendency to veer towards the neuron as the thing, but that's just, I think, a bias because we think of the soma. I mean, if you change your scope and think of, instead of the soma being the important part of a neural unit, the soma might instead just be the life support system that keeps the synapses running, right? And if you think about it in that respect, then the computational work is really being done in the dendrites primarily. And the soma is basically just delivering the decision of the dendrite to some other neurons or some other dendrites, right? Yeah. Okay, that's all I've got to say.
So while I agree with Srikanth that it's very dynamic, but just to stick my neck out, it's fun to be wrong sometimes. There was once I was in a discussion point, and some very interesting discussion came up, and I think that maybe helps us to find some answer to this question. And the idea was that let's ask a question that how many coincident spikes we need to make one neuron spike. Alternatively, you can also formulate it other way around, that how many neurons one neuron can influence, right? So it's basically about one group making one neuron spike, and the next layer also you need perhaps the same number of units to make the next layer spike. So if we think of visual cortex V1, let's say I have some number of neurons active, how many do I need to make something active in V2? after all, this activity has to go from one area to another, to another, to another, to become a sensory motor transformation. So if you think like that, then obviously it becomes a question about how many synapses do you have, how strong the synapses are, and obviously we can then bring the issue of neuromodulation, that how does it modulate the synapses? And the EI balance also comes in, because that will also modulate it. Some very nice experiments done by Mickey London, Peter Latham, and Mike Heuser's group was involved in that. And there they came up with an idea that one neuron adds about 28 spikes. Yeah, something like that. I mean, that's back of the envelope calculation, but it's order of magnitude. There is other similar experiment where Michael Brecht, he stimulates some number of neurons and elicits a behavior in the animal, and their estimate is that perhaps they are influencing 60 to 100 neurons. If we do modeling and we find out, like, if we have those weak synapses, we also need something like 50 to 100 coincident spikes to make a neuron spike. So my money is on that actual functional unit is about that size, given the constraints of synapses and connectivity and the neurons. Yeah, so that's it. So I think that's about it. It's not one neuron, it's not the whole brain, because I mean, obviously whole brain wouldn't make sense. Brain areas perhaps is too coarse. So I think to me that's about, now funnily enough, this number about 50 to 100 can easily coincide with the idea of columns. So
I'd like to agree with what everybody said, which might be, I guess, given what I've said before, the obvious way I would go. But to go back to what I said at the very beginning. So to me, both in terms of, how we think about modeling these things, but also in terms of how we think about functional units in general, a neuron has been the classic functional unit. It's sort of the cell in the brain. It's the things that has synapses and makes connections, and we see it spike. And it's clearly a functional unit. But then if I were to ask, is it the case that if I look at a cortical microcircuit, it's not also a functional unit? If I were to find an appropriate abstraction of that we could parameterize in the way that people have started to think about with hypercolumns in V1, then it too becomes a functional unit. Or if I were to think about sort of cortico-basal ganglia thalamic loops that are processing things, maybe those are also functional units. And maybe, brain areas are coarse, but maybe they're also functional units. The functional unit depends on the level of description for the computation you're talking about. And so to me, I think it's sort of a bit of a misnomer to think that there is one granularity for a functional unit in the brain. Some small set of neurons that are regulating the lobster's somatogastrian system. The neurons are functional units there. They're doing something simple. They're also single or small populations of neurons that are doing very important things in humans. At the same time, in humans, there's probably also large functional units that are doing very interesting and important things at a different level of description. So I would recast the question and ask, where do we have sort of good models that are compact and efficient for thinking about these kinds of functional units? And let's take the neuron as the simple example. We have good models of synapses in the compartmental level. We have good models of sort of active dendrites and the whole neuron. We can reduce that to a Morris-Lacar Fitzhugh-Newman model to sort of capture general spiking properties to make networks. But if I were to ask that question about, say, a cortical microcircuit, something that people have been trying to think about for 20 years, do we have a good reduction of that to something that's more compact, more explanatory, where we have a set of hyperparameters that we can think about as being canonical, but having different variations across the system? And I think we don't. And I think the same thing might be true for sort of the larger loops we see between the basal ganglion cortex and the thalamus, which are clearly conserved, clearly important, and because they have this structure you can see going from posterior to anterior, there's some sense in which they're also functional units. And then brain areas, we know, probably are the same. So all of these, the problem is we just don't yet have a good enough idea of what they're computing, what the right level of granularity to talk about them are, and how to parameterize them in a way that's more compact and efficient. And those seem to be the interesting open questions. In some sense, I think we've got down spikes in a single neuron, to a pretty good first order of approximation. but all of these other levels of the functional unit description are where we have a lot of work to do left.
All right. Well, with that, I want to thank our panel for giving us their time, and I'm sorry that we've gone over by a bit. For those of you who have asked questions that we were unable to get to, I'd like to invite you to please post them on Neurostars, and we will try to answer them there. You could give a label such as week 3, day 1, just to make it clear sort of where they attach to. And with that, again, I'm going to close out the panel. Thank you very, very much, Arvind and Srikanth and Eric.
Sure.
Thank
Thanks.
you.
And thank you all for asking nice questions.
Yes, they were great. We wouldn't be doing this if it weren't for all of you wonderful students.
No, yeah, thanks. May the force be with you.